Someday or the other you might have heard about researches being conducted, which is an essential tool to upgrade ourselves and grow. Today let me guide you through the different components of a research to teach you what are the different features of an experiment. First of all, what is an experiment? We generally have two variables, an independent variable and a dependent variable. IV or the independent variable is the variable that we as a researcher manipulate, and DV or the dependent variable is the one that gets affected when we manipulate the IV. And this investigation of investigating the causal relationship between the IV and the DV by manipulating the IV is called an experiment. Now, on the basis of location, experiments are of two types. One, laboratory experiment, which is a type of experiment conducted in an artificial environment made by the researcher. Now why do we need lab experiments? Now we only want the IV to affect the DV, but in general there can be other variables as well which might affect our DV and change the result. So therefore we must eliminate those variables, and this is done in a lab experiment. The other one is a field experiment, which is just the opposite of lab experiment. It is conducted in a natural environment where we cannot eliminate all the other variables. Now why is natural experiment conducted? It is because there is a term called demand characteristic. In psychological experiments it might be possible. If we take the participants into a manipulated environment, then they might get suspicious and try to behave in a different way and not show their true colors. Therefore we set them in their natural environment so that they don't feel sus and they show their true behavior, which will increase the reliability of our results. Now here comes a new term, reliability, which refers to the extent to which our results are consistent on repetition. Suppose we get our results from the first experiment, then when we and others replicate our experiment other times, then to what extent they are also getting the same results as us. If the results are close enough to ours, then we say our results are reliable, and this happens when there is no demand characteristics such as in a field experiment, and also when there is no extraneous variable acting in our experiments. Extraneous variable is again a new term here that we have encountered. Remember I told you about few other variables which might be present in the environment which have an effect on the DV other than the IV, so a technical term to those other variables would be extraneous variables. After the location, now suppose we have gathered our participants for our research. Let's say you have 30 participants and you divide them into two groups of 15 in each. You have Group A and Group B. This division can be done by different methods. One of the best ways is a way called random allocation, which refers to randomly allocating each participant into groups. There are many ways to do this. One of it could be making small chits of numbers from 1 to 30 written on them, and then asking each participant to pick one from it, and all the odd ones going to Group A and even ones going into Group B. When you have your groups ready, you expose them to the different levels of IV. For example, if our IV is light intensity, so the different levels could be bright, dim, dark. You will three main ways to expose your groups to the levels. The first one is independent measure design, which is exposing each group to a different level for once. Like here you can expose you to bright light and see the effect, and the expose Group B to dim light and see the effect. The other one is called repeated measure design, in which you exposes all the groups to all the levels. For example, here Group A is first exposed to bright light and then to dim light, and in both the scenes we see the effect of IV. Since we are repeating the same group is all the levels of IV, it is named repeated measure design. There is a third type called matched pair design, in which we make pairs of the participants who are similar in nature, behavior, IQ, and other factors. A twin is the best example of this pair. The benefit of this would be that since both the participants have same gene and behavior, all the genetic factors are same, therefore some extraneous variable which might come from changes in their nature and genes are lowered down, which increases the reliability. However, this advantage is missing from the other two. Apart from the two groups who will be exposed to the levels of IV, these groups who are exposed to IV are called experimental group, and other than this, we have a third group called the control group, which is a group which is exposed to a neutral environment in which the IV is absent. You might have heard of control in other science experiments. This is the same. This is done to see if IV is the only factor that is affecting our DV. After the experiment, we compare the results of control to the experimental group and use the control group as a base to compare the experimental group. Now suppose you have a repeated measure design. In this case, there can be general problems among the participants. For example, the very first one is called fatigue effect. For example, if the procedure of an experiment is very long and you make the participants do it again and again, they might get tired because of it and their performance might decrease. Another one is some of them might understand what all they need to do in the procedure, and when they do it again and again they might get a good practice of it. As a result of this, they will be able to do the task very easily, and this would be a problem. Suppose the DV, let's take concentration, is checked by giving the students a test to see their scores. If they are asked to do the same test again and again, they might already know the answer, and they might score really high. If you wonder, why do we need to give them the same test all the time? Why not change it? Then we cannot change it because the test difficulty and questions might also act as an extraneous variable and affect their performance. So we need to keep the procedure of the experiment exactly the same for all the participants in all the levels of IV. This thing of keeping the procedure same in all the different levels of IV is termed standardization of the experiment, which is important to eliminate any extraneous variable caused by the change in procedure. Another problem linked to the same issue is called ceiling effect, which refers to all the participants getting a really high score in a test in all the levels of IV. If all of them got really high scores, then it might also be difficult to interpret if the IV actually caused it, or was it because they got a good practice of the task. Now, one general way to overcome this problem is known as counterbalancing. Suppose if the procedure asks the participants of two groups to do two tasks, 1 and task 2, then firstly we can ask one group to do task in the order of 1 and 2. On the other hand, we ask the other group to do task 2 first, then task 1. This is called counterbalancing, and this is done when dealing with repeated measure design. Thank you for watching.